Even before the mechanisms of innate immunity, which involve cells and tissue mediators, the very first line of defense against external agents consists of a series of prefabricated anatomical barriers, which act as a true boundary between us and pathogens, along with a collection of preformed chemical mediators present in tissues and ready to respond. Before analyzing the barriers the body employs to defend against pathogens, it is essential to understand what these pathogens are, how they cause damage, and in which areas of the body they most frequently reside. Pathogens are microorganisms that have evolved to circumvent the host's defenses more effectively than others, leading to varying degrees of damage. They can thrive in different body compartments with distinct preferences. Extracellular pathogens typically proliferate in interstitial spaces, blood and lymph. This category includes most bacteria, fungi, protozoa and parasites. Others adhere specifically to epithelial surfaces such as Neisseria gonorrhoeae, the causative agent of gonorrhea, a sexually transmitted infection, or Helicobacter pylori, a bacterium with tropism for gastric mucosa. Intracellular pathogens, including viruses and intracellular bacteria, grow within cells. Viruses tend to localize directly into the cytoplasm, while intracellular bacteria, such as the Mycobacteria or Yersinia pestis, the bacterium responsible for plague, proliferate within cytosolic vessels. Another critical distinction among pathogens, which underscores the need for different defense strategies, lies in the mechanisms of damage. There are both direct and indirect pathways. Direct mechanisms include the production of exotoxins, endotoxins, and direct cytopathic effects. Exotoxins, commonly referred to toxins, are proteins secreted by extracellular bacteria, for example, Streptococcus pyogenes or Staphylococcus aureus, whose toxins are responsible for foodborne intoxications. These toxins inflict damage as the innate immune system has limited effectiveness against them. Endotoxins, on the other hand, are structural components of bacteria, not secreted molecules. These are recognized by immune cells, triggering a response that drives the pathology. The most prominent endotoxin is lipopolysaccharide, LPS, a key component of some gram-negative bacteria, such as Salmonella typhi, the causative agent of typhoid fever. The direct psychopathic effect is a feature of many viruses, which, after entering the cell, can directly damage it, for example, by inducing lysis or inhibiting replication. Indirect mechanisms of damage include the formation of immune complexes, the production of autoreactive antibodies, and the activation of adaptive immunity against pathogen-infected cells. Immune complexes are large molecular aggregates formed when multiple antibodies bind to particularly large or highly immunogenic antigens, that is, those presenting multiple epitopes. Immune complexes are a hallmark of any acute infections, but some pathogens are especially proficient at inducing them. For instance, Plasmodium falciparum, the protozoan responsible for malaria, or Treponema pallidum, the bacterium responsible for syphilis, can provoke immune complex diseases. At times, pathogen antigens may closely resemble antigens expressed on host cells, a phenomenon known as molecular mimicry. This leads to the development of cross-reactive antibodies, which target not only the pathogen but also the host cells. A classic example is Streptococcus pyogenes, which shares antigens with myocardial cells. The immune response to this pathogen can result in rheumatic fever, a non-infectious inflammatory disease with potential for severe cardiac damage. Moreover, some pathogens, especially viruses, can induce subtle alterations in host cells, eliciting an immune response against the altered cells. This indirect cytopathic effect is observed with the hepatitis C virus, HCV, which induces non-lethal changes in hepatocytes, affecting the expression of MHC class I molecules. As a result, cytotoxic T cells recognize and eliminate the altered liver cells. The aforementioned mechanisms illustrate how pathogens mount their attacks. Let us now explore the body's initial line of defense, vital for shielding internal compartments and providing the immune system time to mount a response. We can envision our immune defenses as a fortress with multiple layers of protection that become progressively more sophisticated and internalized. Epithelial surfaces represent the outermost barrier of our fortress, offering structural protection. Epithelial cells are tightly joined by occluding junctions, effectively isolating underlying tissues from the external environment. Furthermore, mucosal surfaces, such as those lining the digestive and respiratory tracts, produce a variable amount of mucus, which traps pathogens, preventing them from contracting epithelial cells and facilitates their elimination, often by transporting them to the stomach. A more detailed discussion is warranted regarding the antimicrobial proteins continuously produced by epithelial surfaces and innate immune cells. These proteins remain at the mucosal level, ready to act in response to pathogen invasion. We categorize these proteins into two broad families: antimicrobial enzymes and peptides. Enzymes catalyze chemical reactions that specifically damage bacteria. One of the most important enzymes is lysozyme, found in saliva, tears, and internal fluids, where it is secreted by Paneth cells. Lysozyme binds to peptidoglycan, a structural component of bacterial walls, breaking the bond between N-acetylglycosamine, GLC-NAC, and N-acetylmuramic acid, MUR-NAC, two monosaccharides that alternate to form the peptidoglycan chain. As a result, lysozyme is particularly effective against gram-positive bacteria, which have thick peptidoglycan layers, while it is less effective against gram-negative bacteria, where peptidoglycan is shielded by an outer membrane. Disruption of the peptidoglycan chain compromises the bacterial cell wall, leading to osmotic lysis and bacterial death. A similar but distinct mechanism is employed by another antimicrobial enzyme, secretory phospholipase A2, which penetrates the bacterial cell wall and hydrolyzes the phospholipids of the bacterial membranes, also leading to bacterial lysis. Unlike enzymes, antimicrobial peptides lack enzymatic activity and represent one of the most ancient forms of defense against infections. These peptides are produced in an inactive form as propeptides and are activated by tissue proteases. Key antimicrobial peptides include defensins, cathelicidins, histidins, and lecticidins. Defensins are small peptides, 30 to 40 amino acids, with amphipathic structures possessing both hydrophobic and highly cationic regions. These peptides are highly stable due to a presence of 3 disulfide bonds. Their structure allows them to embed in the bacterial cell membrane, forming pores that disrupt cellular homeostasis, ultimately promoting bacterial destruction. There are several subclasses of defensins based on their amino acid sequences and functions. Alpha defensins are produced by maturing neutrophils and stored in primary granules, lysosome-like vessels that contain substances crucial for fighting infections. Paneth cells in the intestinal crypts also produce alpha defensins, known as cryptins in this context. Cryptins are processed by proteases such as trypsin before being released into the lumen in an active form. Beta defensins differ from alpha defensins in their shorter peptide length and the requirement for cleavage to activate them. Unlike alpha defensins, beta defensins are produced in response to pathogen presence, primarily at sites such as the skin, respiratory tract, and urogenital system. In the skin, they are stored in lamellar bodies, lipid-rich organelles that, when emptied into the extracellular space, impermeabilize the epidermis. In the lungs, beta defensins are present in pulmonary surfactant, a fluid that maintains alveolar integrity and prevents collapse. Finally, theta defensins are unique to primates. Now let's turn to catholycidins. These peptides differ from defensins due to the absence of disulfide bonds. Humans possess only one gene of catholycidins, which is constitutively expressed by neutrophils and macrophages, or induced in response to infections in epithelial cells of the lungs, intestines, and by keratinocytes, which store them in lamellar bodies. In neutrophils, catholycidins are contained in secondary granules in an inactive form and are activated only upon fusion with phagosomes via neutrophil elastase. Structurally, catholycidins include a C-terminal region made up of an amphipathic peptide that disrupts membranes and an N-terminal region whose role remains unclear. Other antimicrobial peptides of lesser significance include histatins, cationic peptides named for their high histidine content. Histatins are predominantly found in saliva and are particularly active against fungi such as Cryptococcus neoformans and Candida albicans. They also promote wound healing. Lastly, we have lecticidins, a family of proteins that fall under C-type lectins. Lectins are proteins capable of binding carbohydrates due to the carbohydrate recognition domain, CRD. Lectins play a pivotal role in immunology, given that carbohydrates form various bacterial structures, particularly in their cell walls. Once a specific bacterium is recognized, lecticidins can kill it directly. In mice, Reg3 gamma is a lecticidin produced by Paneth cells in the gastrointestinal tract. In humans, its counterpart is Reg3 alpha, or HIP-PAP, hepatocarcinoma intestine pancreas, or pancreatitis-associated protein, which, like many of the antimicrobial peptides discussed so far, preferentially kills gram-positive bacteria. In conclusion, the initial defense barriers of our body, although highly elementary, are essential in preventing widespread and potentially fatal infections. Many bacteria are neutralized by these defenses without us ever realizing it, preventing the development of true infection. However, Should these first lines prove insufficient, the body possesses progressively more refined defense mechanisms capable of confronting pathogenic threats.
Thank you for watching. This video was created by School of Biomedical Sciences, Agora. We hope you enjoyed it. If you're curious or have any doubt or question, please feel free to leave a comment below. If you want to find out more about us or want to support our project, click on the following link to visit our website.